Hi, and welcome to this session of regenerative rehabilitation. And before getting started, I would like to thank the organizers for the opportunity to share not only our work, but to represent this emerging field at the summit. I am an assistant professor in the Department of Biomedical Engineering at Oregon Health and Science University, or OHSU. And our group works on the development of engineered therapeutics using translational and rehabilitative animal models for the study and treatment of musculoskeletal traumas. So for the session today, I'll be sharing with you some background on this emerging field of regenerative rehabilitation and how our lab uses these integrative concepts to advance muscle healing. So you'll be hearing from a diverse panel of experts who use regenerative rehabilitation to promote tissue repair and regeneration to treat a broad range of health conditions to address severe unmet clinical needs. So the researchers in this session are brought together through the AR3T, which is the Alliance for Regenerative Rehabilitation Research and Training, and the ICRR, which is the International Consortium for Regenerative Rehabilitation. So AR3T is an NIH-funded resource center that helps to develop research collaborations, provides educational opportunities, and funds pilot projects and technology development projects that will benefit the research community. So this is a multi-institutional network that is anchored by 4 universities, the University of Pittsburgh, Stanford University, the Mayo Clinic, and UCSF. So these universities are the founding members of a larger ICRR, which was formed to bring together leading scientists and clinicians across the domains of regenerative medicine and rehabilitation science in order to drive the creation and transfer of knowledge associated with the development and translation of technologies that restore function and enhance the quality of life of patients. So regenerative medicine focuses on the repair or replacement of tissue lost to injury, disease, or age, primarily through the enhancement of endogenous stem cell function or the transplantation of exogenous stem cells. The focus of rehabilitation science is on the use of mechanical or other stimuli to promote functional recovery. The field of regenerative rehabilitation integrates these 2 approaches, specifically through the application of regenerative technologies with the rehabilitation clinical practices. So this combination has the potential to create new approaches and strategies to treat tissue injury and disease. With a better understanding of regenerative medicine principles, rehabilitation researchers can better tailor rehabilitation efforts to accommodate and maximize the potential of novel regenerative approaches. So engineering the response of cells and tissues to biophysical stimuli has long been the scientific basis for regenerative biomedical engineering. Extrinsic mechanical cues are transmitted through the cytoskeleton and communicate with cytosolic messengers and even the nucleus to regulate gene expression and cellular behavior. So every cell in the body experiences some form of mechanical stimulation, and dynamic mechanical cues are potent modulators of the cellular response and can drive morphogenesis. Mechanical microenvironments include native stimuli such as hydrostatic pressure, shear and compressive forces, and bioelectrical signal. Endogenous cells and tissues experience biophysical forces that naturally guide tissue remodeling, repair, and ultimately healing. Therefore, it makes sense that the application of rehabilitation protocols can promote functional tissue recovery after injury. So following surgery or injury or in the setting of a disease, physical therapy and rehabilitation medicine are often prescribed to accelerate and enhance healing outcomes. This is common practice in the setting of neurologic, neuromuscular, and musculoskeletal injuries, primarily due to the link with mechanical and electrical properties. Rehabilitation can be applied in the form of tissue loading and conditioning, stretching for the application of tensile forces, electrical stimulation, and manual therapies such as joint mobilization and traction, ultrasound, and even the modulation of tissue temperature by the application of heat or ice. In some cases, prehabilitation or the implementation of exercise programs prior to an injury, for example, in the case of an elective surgery, can serve to prime the tissue response and optimize repair capacity. The key concept that guides regenerative rehabilitation is the idea that innate regenerative processes can be driven by biophysical, electrical, and thermal stimuli. The underlying concept is that these stimuli can guide the regenerative potential of transplanted and endogenous cells to optimize functional recovery. A primary focus of regenerative rehabilitation research is to elucidate the cell-cell and cell matrix interactions that are influenced by these stimuli and to engineer an interaction that will result in improved functional outcomes. The talks that you will hear today will give a full breadth of ex vivo and in vivo approaches that look to modulate the environment at the subcellular level all the way up through the full body. So as the first research talk in this regenerative rehabilitation session, today I will be describing our combinatorial biomaterial-based approaches for regenerating skeletal muscle and how we can combine 3D scaffolds that exhibit nanoscale patterning with rehabilitative exercise to enhance the host response to injury. So the skeletal muscle is quite a privileged organ in its remarkable ability to self-repair. In contrast to other tissues, injured skeletal muscle has an intrinsic capacity to adapt and initiate a synchronized biological response to prevent further muscle loss and eventually lead to regeneration. Minor injuries such as muscle strains and contusions will heal spontaneously in healthy adults. The process of normal healing in response to minor injuries involves the activation of resident muscle stem cells or satellite cells, which normally reside in a quiescent state on the muscle myofibers. However, following injury, they become activated and begin to proliferate and undergo changes in their cytoskeletal organization and shape, entering a precursor stage as a myoblast or myocyte. Within a few days, these myocytes then fuse to form multinucleated myotubes that further mature into myofibers, which are the primary contractile units of skeletal muscle. But what we want to explore is what happens when these native programs don't work as intended or simply cannot outpace fibrosis. In some cases, the injury to the skeletal muscle is so extensive that the normal healing programs cannot regenerate the damaged tissue, and in place of new myofibers is non-functional fibrotic scar tissue, a condition known as volumetric muscle loss, or VML. So VML injuries consist of extensive fibrosis within the injured muscle and is associated with a reduction in muscle strength and joint range of motion. So current approaches for treating VML in the clinic include scar tissue debridement, which involves the removal of non-viable or infected tissue and any foreign materials. Debridement is often coupled with free functional muscle transfer, in which muscle from another part of the body is excised and transplanted to the site of injury. Despite advancements in limb salvage efforts, the mediation of VML injury is associated with extensive donor site morbidity, graft failure, and may be limited by the availability of healthy muscle. One regenerative approach that has the potential to circumvent these challenges is the application of off-the-shelf biomaterials that possess the ability to restore both tissue, structure, and function. So given the complexity of biological tissue, strategic biomimicry through the thoughtful pairing of cells with bioactive scaffolds and biomechanical environments enables recapitulation of the physiologic structure-function relationship and advances the pathway towards clinical translation. To regenerate skeletal muscle, we can look at the template that nature has provided. So muscle tissue is composed of highly aligned myofibers that coalesce into functional units. Alongside the myofibers is an extensive vascular network to support the high oxygen consumption by skeletal muscle. In terms of the native composition of skeletal muscle, the tissue contains a wide range of collagens, with the predominant collagen type in skeletal muscle being type 1 and type 3, which align into an array to form fibrils in the tissue. Hence, we decided to use collagen type 1 as our primary biomaterial for skeletal muscle regeneration. Some of the advantages to using collagen is that it possesses a stiffness range that is close to muscle, has the ability for the incorporation and release of growth factors, allows for architectural and spatial control, and possesses the natural ligands for cell adhesion. We have previously reported the fabrication of patterned collagen scaffolds with nanoscale features that are capable of directing and guiding cell cytoskeletal organization. These scaffolds are fabricated using a shear-based extrusion approach in which monomeric high concentration collagen is extruded at a high velocity into a neutral buffer to initiate fibrillogenesis along the direction of the extrusion, creating a highly aligned and highly oriented fiber orientation that is on the order of around 30 to 50 nanometers in diameter. And similarly, we're able to create randomly oriented fibers with the same material, but by slowing down the extrusion process. So when seated with cells, these scaffolds guide the cell cytoskeletons along the direction of the fibers. And we have demonstrated this modulatory capacity with a wide range of cells, including ECs, iPSC ECs, cardiomyocytes, fibroblasts, muscle cells, and myogenic cells. So the ability to control myogenic organization bodes well for myogenesis, in which individual myogenic precursors need to fuse end-to-end to create elongated multinucleated myotubes. So on our scaffolds, we have shown that the pattern topography guides myogenic differentiation and fusion efficiency, as well as maturation of myotubes in vitro. We've also shown that topography-dependent improvements in maximum contraction velocity and contraction area were seen upon electrical stimulation, as well as increased angiogenic and myogenic cytokine production and gene expression. So in order to assess the therapeutic potential of our scaffolds in a muscle injury model, we used a mouse model of VML, or volumetric muscle loss. So the pathological features of VML were recapitulated by a surgical ablation using a microblade of a defined mass from a TA muscle. So we removed about 20% of that TA muscle. And we have representative images here of H&E staining of a whole TA muscle cross-section that shows the injured TA muscle on the left and a TA muscle 30 days following VML injury on the right. And a dashed line indicates the area corresponding to the muscle area that has been removed during the surgical procedure. So the injury does not heal on its own and is associated with peripheral fibrotic scarring and reduced muscle mass and forced contraction. In subsequent studies, we transplanted cell-seeded pattern scaffolds into the site of injury in our VML mouse model to assess muscle regeneration and graft integration. We demonstrated improvements in myogenesis and angiogenesis when aligned pattern scaffolds were transplanted with myogenic endothelial cells compared to scaffolds with randomly oriented fibers, highlighting the importance of scaffold topography in modulating tissue regeneration. However, a key limitation of this approach is cell source availability, which hinders the translational feasibility of this method. So one approach involves the integration of advanced orthopedic surgical care with comprehensive physical rehabilitation programs, which have produced salient advancements in returning injured patients to the highest level of function and quality of life. So there are many benefits to a well-crafted physical rehabilitation regimen. So physical exercise stimulates vascular flow, and angiogenesis, it regulates the activation of muscle satellite cells and stimulates ECM remodeling. Additionally, cyclic mechanical stress induces the production of myogenic growth factors such as IGF-1. So the IGF-1 gene is normally activated in response to mechanical stimulation produced by repeated cyclic stretch, and without normal cycles of tension and stretch, this pathway sits inactive, leading to muscle atrophy and thereby underscores the importance of physical exercise in muscle health and healing. So single intervention acellular therapies can be paired with a mild to moderate rehabilitation regimen that can offer the potential for a more streamlined recovery. Therefore, we hypothesized that an exercise regimen combined with an acellular pattern scaffold could provide a translational and off-the-shelf regenerative approach for the treatment of VML. So in the design of our study, it was important that certain aspects of the translational feasibility be considered. For example, our scaffold is acellular and exhibits the same aligned topography that we had previously demonstrated to enhance myogenesis and angiogenesis. And secondly, we incorporated exercise in the form of activity running wheels to stimulate blood flow and satellite cell activation. However, many patients are unable to exercise immediately following injury, and in fact, pilot studies showed negative remodeling and fibrosis in our VML mouse model if exercise was introduced too early. Therefore, we used an exercise regimen that begins 7 days following initial VML injury and lasts for 14 days for a total of 21 days from injury to final assessment. However, this first week is a critical time for healing. Therefore, to influence the homeostatic programs initiated during the initial rest period following injury, delivery of IGF-1 served to bridge this critical time during which the exercise-induced healing response is limited. So our pattern 3D scaffold was fabricated as in our previous studies and shown here on the left by SEM imaging and highlights the aligned orientation of the collagen fibers. A cumulative release of IGF-1 over the course of 21 days was quantified by ELISA, showing a total release of 1,250 nanograms of IGF-1 with a burst effect occurring within the first 24 hours. So to test the bioactivity of IGF-1 releasing scaffolds, the myotube formation capacity of mirroring myoblasts was quantified. On IGF-1 releasing scaffolds seeded with myoblasts, the resulting myotubes formed were 1.5-fold longer in length and contained 2-fold greater nuclei per myotube when compared with scaffolds without IGF-1. When implanted into the ablated TA muscle, we observed greater de novo formation of myofibers, a 2-fold increase in the abundance of mature neuromuscular junctions, and a 3-fold increase in the density of perfused microvessels in animals that were treated with IGF-1-laden scaffolds, coupled with exercise, compared to control scaffolds that were transplanted into mice that did not receive exercise. So this suggests the modulatory role of both IGF-1 and exercise in these regenerative outcomes. So in summary, in the current study, the spatial patterned aligned topography of the engineered scaffold served to control cytoskeletal interactions through nanoscale guidance cues that directed cell orientation and organization along the longitudinal anatomy of the muscle. Incorporation of IGF-1 into scaffolds served to stimulate proliferation and initiation of myogenic fusion of skeletal muscle myoblasts in vitro, and likely aided in the activation of resident stem cells and recruitment of endogenous endothelial cells to the scaffold and injury site. Taken together, we have shown that through the combined transplantation of patterned biomaterials with mechanical stimulation in the form of voluntary running exercise, that large volumetric skeletal muscle defects in mice can be regenerated and reinnervated to an extent that is comparable with native tissue. So to bring everything full circle, exercise programs can stimulate the donor-host interface to foster integration and promote a series of beneficial regenerative responses. The combination of rehabilitation protocols with cellular therapeutics can enhance transplantation efficacy and synergize with the host response to guide beneficial tissue remodeling and repair. However, excessive loading following injury or implementation of exercise too early in the recovery process can trigger a cascade of deleterious consequences that worsen tissue dysfunction and impede recovery. Therefore, a better understanding of the synergistic effects of rehabilitation approaches on tissue regeneration will be pivotal in the clinical development and implementation of functionally relevant treatments. Overall, this is a tremendous there's tremendous potential in the emerging field of regenerative rehabilitation to guide the development of targeted clinical rehabilitation programs that can permanently change the way we approach regenerative medicine. And so to end with, thank you to all of our collaborators back at Stanford and to our new lab members here at OHSU, especially to Cynthia Alcazar, who played a critical role in the design and application of our translational animal injury models. And also thank you to our funding sources and again to the organizers for the opportunity to share our work.